In the last video, I had discussed about outbreeding devices. Now let us see the next topic, that is pollen pistil interaction. Now you are aware as to what is pollen and what is pistil. Pollen is produced inside the anther, and this acts as a male gametophyte, pollen grain. Whereas when we talk about pistil, it has three parts mainly: the stigma, style, and the ovary. Now over here we are talking about the pollen. When it reaches onto the stigma of the pistil, both of them have to exchange some kind of dialogue so that they can take part, whether the pollen grain can germinate and take part in fertilization or not. That will be decided by what we call over here as pollen pistil interaction. Now here, if the pollen is of right type, then this will be termed as compatible, and if it will not be of a right type, then that will be referred as incompatible. तो फर्टिलाइजेशन कब होगा? जब वो compatible होगा, means after reaching onto the stigma, both of them are going to interact, and then recognition will take place. Okay, recognition will be done by the stigma, and then later on it will be decided whether it is going to take part in fertilization or it is going to be rejected. अगर एक्सेप्ट किया तो फर्टिलाइज हुआ, पहले पहचान होगा, एक्सेप्टेंस होगा, और अगर मान लो एक्सेप्ट नहीं किया गया, if it is not compatible, then that is going to be rejected. As a result of that, fertilization is not going to take place. The pollen grain is not going to be allowed to germinate, not allowed to make its or develop its pollen tube in order to carry the male gamete to the or towards the ovary. Now here often what happens, pollen grain of wrong type also reaches onto the stigma. Means what? Wrong type over here we can refer as what? Pollen of different species can reach onto stigma of another type of species or another species, then that will be called as incompatible. Earlier topic we also have discussed about self incompatibility. Self incompatibility means what? Sometimes what happens, this is a genetic process, or otherwise this is a genetic mechanism in which what happens, the pollen grain produced from the same plant is not accepted by the stigma. So as a result, that becomes self incompatible, and so it is not allowed to germinate, not allowed to extend its pollen tube and grow towards the ovary for fertilization. Now here the pistil is able to recognize, earlier I mentioned, it is able to recognize the pollen, and if whether it is of right type or a wrong type. Right type है तो compatible, not right यानी wrong है तो it will be called as incompatible. Means the pistil has this ability. Now if it is of right type, then the pistil is going to accept the pollen. It is going to promote the further events, that is post pollination events are going to be followed, or that is going to proceed further, and that is going to lead to fertilization. And if the pollen grain is not of right kind, means it is of wrong kind, मतलब क्या हुआ? Incompatible, then the pistil is going to reject it, and as a result of that, the pollen germination is prevented on the stigma. And if at all, if it is going to germinate, then further the pollen tube growth inside the style so that it can come down towards the ovary is going to be prevented. Now this ability of the pistil, to recognize the pollen, and then later on whether it is going to be accepted or it is going to be rejected, that is a result of a continuous dialogue between pollen grain and the pistil, as I mentioned earlier. And this dialogue is mediated by some chemical components. What we have studied earlier about chemotropism, Okay, chemotropism. So that over here is playing an important role. Now the botanists have been able to identify in the recent years some of the pollen and pistil components. Some of the pollen pistil has been identified by the botanist, and they have now understood the interaction between both that leads to recognition and then later on either by acceptance or otherwise by rejection. मतलब पहले पहचान किया पिस्टिल ने, उसके बाद उसको या तो एक्सेप्ट किया या रिजेक्ट कर दिया. एक्सेप्ट किया तो फर्टिलाइजेशन होगा, रिजेक्ट किया तो फर्टिलाइजेशन इज गोइंग टू नॉट गोइंग टू टेक प्लेस. नाउ लेट अस से दैट आफ्टर द पोलन ग्रेन इज एक्सेप्टेड, मतलब ये हुआ कि वो कंपेटिबल है. द पोलन ग्रेन हैज टू जर्मिनेट on the stigma and produce a pollen tube, and this pollen tube has to germinate from the, or otherwise has to come out of the pollen grain through the germ pore. If you remember earlier we have discussed about the structure of a pollen grain. Pollen grain is composed of two, or rather bounded by two layers, two wall layers. Outer layer is exine, inner layer is intine. Intine is a continuous layer inside which a plasma membrane is present. Whereas the outer layer exine, this is not continuous. At different locations there are spaces left there, means exine part is not there. And if you remember sporopollenin के बारे में बात किया था. Sporopollenin is absent at these locations which are called as germ pore. So from these germ pore, what happens? The pollen tube germinates and starts growing bigger in size, during which or through which what happens? The two nucleus we had talked, or otherwise two cells we had talked, vegetative cell and one more was there if you remember, generative cell. Vegetative cell and generative cells are found here. Now both of them are going to move with the growth of the pollen tube. Inside the pollen tube they are moving. Now whatever is present here inside the pollen, it is going to move here. So here if you see in this point it is given that even the content of pollen grain move into the pollen tube. The pollen tube grows through the tissues of the stigma and the style and reaches the ovary. Earlier in this figure also it was shown, so many pollen grains are found here, but one of them is shown to elongate and reach to the ovule down here inside the ovary. Now coming here further, if you remember, just now I have discussed, but if you remember we have talked about the pollen grain, that 60% of the angiospermic plant, or in 60% of the angiospermic plant, what happens? The pollen grain is shed at a two-celled condition, and these two cells are what? Vegetative and generative cell. And then later on, when this pollen tube, let us say that pollen tube has germinated now, it has come down. In this what happens? These two cells start moving down, and while they are moving down here, almost they have reached towards the ovary over here, or inside the ovary towards the near to the ovule. At that time what happens? One of the cell, that is the generative cell, okay, that is the generative cell, is going to divide again by mitotic division. As a result of that, two sperm nuclei, or otherwise two male gametes are produced here, and this tube cell, also called as vegetative cell, is going to disintegrate before entry of the male gamete inside the ovule, and that entry will take place through the micropyle. Now in this, okay, and then other plant, or we had talked about around 40% of the angiosperm plant, the pollen grains are shed at a three-celled condition. Means what? It has a vegetative cell, and generative cell has already divided, as a result of which two male gametes have been created. Okay, so that is two male gametes have been created and plus one vegetative cell. So that's why three cells are already present inside the pollen grain. So as a result of germination of pollen grain and extension of the pollen tube, that is growth of the pollen tube, these three cells, that is one is the vegetative and two sperm nuclei or otherwise male gametes are going to move down towards the ovary here. Now coming down, the pollen tube, after it reaches to the ovary. Now after it reaches to the ovary, now if you remember in the in the embryo sac, we had studied about 7 celled and 8 nucleated. Out of that 7, 3 are arranged near the micropylar end, 3 are arranged towards the chalazal region or at chalazal region, and one cell, large cell, is there in the center which is having 2 nuclei which is called as polar nuclei. Now when the pollen tube has reached to the ovule through the micropyle, now this pollen tube has to enter, and at that time down over here in the figure, if you note, down over here 3 cells are there, 2 synergid cell and 1 egg cell. Now this synergid cell we had discussed are having an apparatus which is called as filiform apparatus. So what happens, this pollen tube enters into the embryo sac through one of the synergid, okay, through a filiform apparatus. Now this filiform apparatus, उसका काम क्या है? Okay, it guides the entry of pollen tube. उसका काम यही है कि pollen tube को guide करना है, so that pollen tube can enter and release the 2 male gametes. Now one male gamete has to fertilize or fuse with the egg nucleus, and the other one has to come and fuse with the 2 polar nuclei inside the large polar cell और सेंट्रल सेल. लार्ज सेंट्रल सेल के पास 2 न्यूक्लियस है, उसके साथ फ्यूज करना. सो एज अ रिजल्ट 2 न्यूक्लियाई इफ यू जस्ट टेक एज n, so this is n plus n, and 1 nucleus has come from male gamete, so n plus n plus n, 3n. So that is going to develop as 3n, which is called as now PEN, primary endosperm nucleus. Primary endosperm nucleus, which is triploid. This is also called as triple fusion. This is also called as triple fusion. And the other one earlier, the first is what? Fusion with egg nucleus. Fusion of 1 male gamete with the egg nucleus results into development of a zygote. Now this zygote is going to develop into embryo later on. Whereas what we discussed just now, this polar nuclei 2 are there, they fuse with the second male gamete and form 3n, which is called as over here as PEN. That is going to develop as endosperm, which is going to develop before the development of embryo, so that it can provide nourishment to the embryo. Now coming here, as we have earlier discussed, that the process of recognition then followed by promotion or inhibition, that is acceptance or rejection, all these, that is interaction between pistil and the pollen, is a dynamic process. It is a dynamic process. Now all the events starting, that is all the events from the pollen deposition on the stigma until the pollen germination, pollen tube elongation towards the ovary, all these are referred as pollen pistil interaction. Now this is very important. This is very important for a plant breeder to gain the knowledge or to have the knowledge, so that the plant breeder can manipulate or otherwise use that knowledge in manipulating the pollen pistil interaction. Even in condition of where the pollen grain is incompatible to the stigma, so that they can get a desired hybrid, they can develop a hybrid plant. So that is, I'll continue this. in the next video.